In this video, we are going to review the process of preparing a bacterial smear for staining and the gram stain process. We are going to prepare our smear from culture B, which we will use in Prac 1. Culture B has yellow and white colonies present, indicating two different types of bacteria, so we need to create smears and gram stain both. We begin by gently passing a clean slide through the blue flame to warm it and clean off any residue. We then use our wax pen to label the ends of the slide, Y for the yellow colony and W for the white colony, as we are going to smear the two different colonies on the same slide. Next, we need to collect water to create our smear. We do this using our loop, turning on the tap and then immediately turning it off. We can then catch the drip hanging from the tap and give a gentle flick to remove the excess. We then spread the water on one end of our slide. We repeat the process so we have another drop of water at the other end of our slide for the other colony. Now we need to sterilise our loop so we can pick colonies from the culture plate. Before we pick the colony, we cool our loop on a clean part of the agar, and then just touch our loop to one colony, in this case the yellow colony. We don't want too much bacteria on a slide, so we don't even need to pick the entire colony. Then we spread the colony in the water on our slide as much as possible without touching the other water drop at the other end. We sterilise our flame again and repeat the procedure with the white colony. Cooling our loop on a clean part of the agar, and then just touching the white colony. And spreading it on the slide. We sterilise our loop again to clean it before setting it aside, and then allow our slide to air dry completely before heat fixing. If the slide is not completely air dry before heat fixing, the bacterial cells will be damaged by the heat. We heat fix the bacteria by passing the slide through the blue flame 3 times. We are now ready to perform a gram stain or any other type of bacterial stain. Once the smear is dry and heat fixed, we no longer need to maintain the sterile space, so we can switch off the flame for the staining process. Laying our slides on a staining rack over the sink, we start by flooding the slide with crystal violet. And letting it sit for 1 minute. Next, we wash our slides in tap water. Make sure the water flow on the tap is slow and gentle, and rinse your slides thoroughly. Tipping off any excess water, we then flood the slides with iodine, which will mold the dye in the gram-positive bacteria. We let the iodine sit for 1 minute as well. Again, we gently wash the iodine off our slides in slow flowing tap water. After this, we apply the alcohol decolorizer. Start the water running before you begin this step, so you can wash as soon as you need to. Then, gently drip the alcohol over the slide for a few seconds, watching the runoff. As soon as you stop seeing purple dye in the runoff, immediately wash the slide in water. The final step is to counterstain the slide with gram-safranin. This will counterstain the gram-negative bacteria. Flood the slide in the safranin and let it sit for 1 minute. Then again gently wash the slides in tap water. After the final wash, gently blot the slides dry with paper towel. Your slides are now ready to be viewed with a microscope. You do not need to add a cover slip for these slides.